So my apologies, everybody, for that little technical glitch. But it's, as I say, a real pleasure to be able to join the meeting today. And I'd like to thank you for the invitation and for a great topic to discuss, actually, and a great topic for an oncologist from the UK to talk about as well. So these are my disclosures. And I thought it would be good to start with a plan of the presentation. And as Pete mentioned, I'll discuss a little bit the data that we have available for off-label use of various drugs in GIST, including Nilotinib, Everolimus, Dasatinib, Pazopanib, Sorafenib, BRAF inhibitors, and also immunotherapy. But in many ways, you know, this is not an exhaustive list, and this is very much an ongoing discussion between patients and oncologists. And I think we're in a very fortunate position right now to have lots of good options available for patients with GIST. And it's a question of finding the right fit for each individual person. And I think I'd like this to be more of a discussion piece rather than me sort of just talking about individual drugs and the data to support individual drugs. The second component involves a brief discussion about NTRK inhibition in sarcomas and in GIST. And as Pete mentioned, this is a very exciting field, particularly in the context of a so-called tumor agnostic approval for NTRK inhibitors. And then thirdly, just a sort of brief review of the current and upcoming clinical trials in GIST. I'm aware that other speakers have discussed this a little bit, but again, I hope that this can be more interactive with lots of questions and discussions at the end of the talk. So I prefer it to be very, very interactive in that respect. So as we all know, there are a number of drugs approved for treating GIST: Imatinib, Sunitinib, Ripretinib, more recently Ripretinib, and also Avapritinib for tumours that harbour the D842V PDGFR alpha mutations. So that's a good starting point. And I think in many ways, because there are lots of good options and more options becoming available, the question regarding off-label use becomes sometimes less relevant depending on which country you're in. So in terms of off-label use of systemic therapy, there are factors that involve where an individual person lives. Of course, in certain countries, such as the United States and Germany, it is easier to obtain access to treatments that have been approved for other indications. I would say in countries like the United Kingdom that can be a little bit more challenging, but nothing is impossible. And it is sometimes feasible to get off-label use for individual patients. But again, this is in the context of the number of drugs that are already approved for GIST. And in a way, following on from Ramesh's presentation, that toxicity management is really important to enable continuation of a therapy. And we have many people in clinic that have been on Sunitinib and Ripretinib, for instance, for a very long time, with essentially an attenuated or hybrid schedule where we've adjusted the dose, adjusted the schedule, depending on the side effects that they experience, and they've been able to carry on with the tyrosine kinase inhibitor despite initial toxicity, maybe on the approved dose. So I think that that's very important to mention in this talk as well. For a number of our patients in clinic, we have access to compassionate use and expanded access programs. I suppose this now primarily applies to Ripretinib and Avapritinib. And following on from Ramesh's presentation, multidisciplinary approach is really, really important. So, for instance, if there is solitary progression in one site, then local therapies can be used to deal with that site of progression to enable people to continue on a tyrosine kinase inhibitor for longer. So all of these things are really important to try and get the best out of each individual drug. And of course, clinical trials are very, very important, and the only way that we'll really get to move things forward in terms of regulatory approval, etc. And we're very fortunate at the Royal Marsden to have access to a phase 1 program as well, which gives us access to novel drugs, which again, for a patient with a specific type of mutation or a specific type of GIST could offer access to drugs that they wouldn't otherwise necessarily be able to receive. I think in terms of off-label use of systemic therapies, it's important to have a very careful discussion between the patient and the oncologist. I often discuss with patients the option of a clinical trial. Would a clinical trial at the present time be a better fit for that individual person? Will drug supply and drug availability be a question in the future? And what are the sort of side effects and the pros and cons and the available evidence that we have for the use of this particular drug in this particular indication? And of course, this discussion changes with each individual person, and is a discussion to be had with patients if we're considering off-label use of any systemic therapy for any type of cancer. I think for this presentation, I'd like to focus on the available clinical data and experience for individual drugs that have never been approved for GIST, but clearly do have some form of activity. And basically, I think we have to be pragmatic in our approach, and as Ramesh highlighted, work together to find the best solution for each individual person. So Nilotinib is a well-established tyrosine kinase inhibitor in the treatment of chronic myeloid leukemia. Because of the targets of this drug, there's also been interest in its use in GIST. And a few years ago, a randomized phase 3 trial was published, which basically randomized nearly 650 patients to either receive Nilotinib or Imatinib as first-line therapy. And in terms of the progression-free survival, the median progression-free survival for Imatinib was 29.7 months, and for Nilotinib, 25.9 months, with a hazard ratio of 1.47. There was some differential activity in terms of the mutational status, as shown on this slide. For tumours harbouring the KIT exon 11 mutation, the median progression-free survival for Imatinib was 32.3 months, and for Nilotinib, it was not reached. In terms of the exon 9 population, the median PFS for Nilotinib was only 3 months and not valuable in the Imatinib arm. But you can see from the confidence interval that that there was a significant benefit there for Imatinib. In terms of the side effects, they were relatively similar to many other tyrosine kinase inhibitors in terms of anemia, blood enzyme changes, and abdominal pain. There's been a phase 1/2 trial of the mTOR inhibitor Everolimus in combination with imatinib, and this trial included 2 strata. The first for patients who had had disease progression on imatinib, and this included 28 patients, and the second for patients that had had disease progression on imatinib plus sunitinib or another tyrosine kinase inhibitor. And this stratum included 47 patients. And the median PFS for the first stratum was 1.9 months, and for the second, 3.5 months. And in terms of common adverse events with the mTOR inhibitors, so diarrhea, nausea, fatigue, and anemia are all relatively common side effects with this type of drug. And this slide here from that publication shows the sort of rationale for using a combination of drugs targeting this signaling pathway, including mTOR and PI3 kinase inhibitors in GIST. So basically using these in combination with imatinib. I would say that so far the results have been promising, but I think we probably need to find other options and other combinations and other drugs targeting this pathway to improve these outcomes, as illustrated on this slide. Another drug that's been investigated in a number of prospective trials in GIST is Dasatinib. The Chinese group recently published a phase 2 trial of 58 patients. And in this phase 2 trial, the 3 months progression-free survival was 53%, with a median overall survival of 14 months. And they reported that the primary nor secondary gene mutations predicted the efficacy of this drug in this cohort. And common side effects with this drug were anemia, proteinuria, fatigue, neutropenia, and diarrhea. Similarly, the Swiss group have performed a phase 2 trial of Dasatinib in GIST and used FDG PET CT to assess response to treatment, with very good response rates using that modality, with a median progression-free survival of 13.6 months, and the median overall survival was not reached. So very promising data from this phase 2 trial. The drug was relatively well tolerated. In 5% of people, there were grade 4 toxicities, so grade 4 adverse events, serious adverse events, and in 48% of patients, so grade 3 toxicities, and most of these affected the gastrointestinal tract or the lungs. And the dose was interrupted or reduced in 25% of cycles. And again, this highlights that a lot of these drugs need close monitoring in terms of toxicity management and in terms of dose reduction and schedule adjustment. In addition, we were able to obtain off-label Dasatinib for one of our patients, which is unusual for the UK. But this little slide illustrates the benefit that this can actually provide for individual patients. So this gentleman was diagnosed with a locally advanced rectal GIST back in 2012, and because of the anatomic position of this tumor, it was deemed to be inoperable, or if an operation had been performed, it would be life-changing, essentially. And he was therefore commenced on imatinib, and initially had a good response that stabilized, but the following year we changed treatment to Dasatinib, 50 milligrams twice daily, and he had an excellent additional response to Dasatinib. He did have some toxicity from this agent in terms of a pleural effusion, and we gave him a treatment break and then restarted the drug. And he continues to tolerate this drug very well, and essentially has had a complete response to treatment. So his most recent scan has shown no real evidence of disease, just a plaque of a change on the scan where the tumour used to be. So again, just highlighting that for individual people, off-label use can have a profound effect in terms of management choices. There's been a lot of interest in Sorafenib as well in GIST. So this phase 2 trial recruited 31 patients, and the response rate was 13%. 52% of patients had stable disease, median progression-free survival of 4.9 months, and median overall survival of 9.7 months. And the drug was relatively well tolerated in this trial, and reported similar and well-known side effects for Sorafenib, so hand, foot reaction, fatigue, high blood pressure, and abdominal pain. And I've had patients that I treated in the United States, just patients treated with Sorafenib off-label that could continue on the drug for a long time, but with careful management of side effects and, as I say, dose adjustment and schedule adjustment, which is absolutely critical with all of these drugs. There have been a number of other retrospective studies that have shown, again, benefit for Sorafenib. In this retrospective series, the response rate was 5%, and 52% of patients had disease stabilization for greater than 4 months, with a median progression-free survival of 7.7 months and median overall survival of 13.5 months. And again, the similar side effects, which are well established for this drug in terms of diarrhea, hand-foot reaction, fatigue, loss of weight, and skin reactions. So Pazopanib is another tyrosine kinase inhibitor that's approved for soft tissue sarcomas, and again, probably an easy drug to obtain access to for GIST patients off-label. There have been 2 trials published. The first by Dr. Ganju and colleagues was a phase 2 trial that recruited 25 patients. The best response in this trial was stable disease. Median progression-free survival was only 1.9 months, and median overall survival was 10.7 months. And Pazopanib in this trial was reasonably well tolerated, with no new or unexpected toxicities. The French sarcoma group have also performed a randomized phase 2 trial of Pazopanib in GIST, randomizing 40 patients to receive Pazopanib, and 41 to receive best supportive care. Basically, they observed that there was a significant benefit for Pazopanib in this trial, with a median progression-free survival of 3.4 months compared to 2.3 months for the best supportive care group. And the patients that were on best supportive care alone and then switched over to Pazopanib had a median progression-free survival from starting Pazopanib of 3.5 months. And similar to previous trials of Pazopanib in soft tissue sarcomas and renal cancer, the toxicity profile was similar, with things like hypertension, leukocytis, tiredness, diarrhea being common side effects, but these are well-known side effects of this drug. One of the other things that's really important, and Pete mentioned this earlier, is that you can describe the use of a drug as off-label, but it's actually off-label for the indication, but it's on target for the particular tumor that we're trying to treat. And the use of BRAF inhibitors in BRAF mutated GIST is a clear example of this principle. And again, a clear benefit for patients in terms of off-label use of these drugs. There's been a lot of interest in immunotherapy for many types of cancers, including melanoma and non-small cell lung cancer. Clinical results so far in terms of immunotherapy in sarcomas and in GISTs specifically are limited. Dr. Singh and colleagues have presented data on a randomized trial of the checkpoint inhibitor nivolumab, either alone or in combination with the CTLA-4 inhibitor ipilimumab. And patients who took part in this trial had received at least imatinib previously. 29 patients were recruited. 27 of these patients were valuable for efficacy. And in the nivolumab alone arm, 7 out of 15 patients had stable disease as best response, with a median progression-free survival of 8.57 weeks. And then in the combination arm, 1 out of 12 patients had a partial response and 2 had stable disease, with a median progression-free survival of 9.1 weeks. Notably, 8 patients on this therapy were on treatment for greater than 6 months, and 2 patients with a KIT exon 17 mutation had tumor shrinkage on imaging. And the most common adverse events were fatigue. Higher grade adverse events occurred in the combination arm, but also in the single agent arm. And these included high glucose, weakness, diarrhea, as well as rash and fatigue. At the Hutch, we actually treated GIST patients with another checkpoint inhibitor, nivolumab, off-label. This young woman had highly refractory disease and had KIT PDGFR wild type. She basically presented in 2000 and had been on active surveillance for a number of years. She was treated with imatinib, sunitinib, and regorafenib, also treated within a phase 1 trial, as well as with sorafenib off-label. Unfortunately, on sorafenib, she developed skin reaction, hand-foot syndrome, and was very reluctant to try another tyrosine kinase inhibitor. And given the context, we obtained compassionate use nivolumab, and she had a durable response to treatment and tolerated the drug very, very well. Again, highlighting the importance of off-label or compassionate use access for GIST patients. So moving on to NTRK inhibitors. Tropomyosin receptor kinases are receptor tyrosine kinases encoded by 3 genes, neurotrophic tyrosine receptor kinase 1, 2, and 3. And these function as high affinity receptors for neurotrophins, which have an important role in the development and normal function of the nervous system. Some lesions have been identified, particularly in pediatric sarcomas, and most notable being infantile fibrosarcoma. But in terms of other sarcoma subtypes, we currently have very little knowledge which are most likely to harbor TRK fusions. But even though the number of sarcomas that may have tract fusions is low, the response rates that have been published and the durability of these responses to NTRK inhibitors is very impressive. And this has led to a histology agnostic approval for drugs such as Larotrectinib in tract fusion positive cancers. And this is the first reported response in a patient with metastatic sarcoma, so very aggressive, high-grade undifferentiated sarcoma. And you can see the stunning response to Larotrectinib on this slide. Also, with another NTRK inhibitor, Entrectinib, again a very impressive response to treatment. And there have been investigations into how frequent these fusions are in GIST, and Jason Siklik and colleagues have published this study looking at GIST patients, including KIT PDGFR alpha wild type tumours. And there have been very good responses to treatment with NTRK inhibitors in patients with tumours harbouring NTRK fusions, as illustrated on this screen. So you can see the sort of dramatic response here on PET-CT, with a reduction in FDG uptake, as well as a decrease in size of the tumour. And at 4 months, the patient had an ongoing partial response to treatment. So again, highlighting the importance of tailoring treatment to an individual patient and tailoring the treatment to the characteristics of the tumour. Recently, there have been a recommendation paper that we published in Annals of Oncology this week on the diagnosis and management of tract fusion sarcomas. Again, we have a lot to learn about these fusions in various sarcoma subtypes, but this guideline tries to present a schema for testing NTRK in sarcomas. And as you can see in one of the figures from the publication, that some of the pediatric sarcomas that I mentioned, infantile fibrous sarcoma, high frequency of NTRK fusions, but some of the other sarcomas, including KIT PDGFR alpha wild type GIST, may have a lower frequency, but well worth testing for NTRK status. Then just quickly moving on to clinical trials. Ripretinib, of course, was approved by the FDA as fourth-line therapy this year, based on a placebo-controlled phase 3 trial with a median progression-free survival of 6.3 months for Ripretinib versus 1 month for placebo. I think this drug is very well tolerated. Our patients have done really well on this drug, and some of whom have been very heavily pre-treated. And of course, there's a second-line trial running currently that randomizes patients to receive either Ripretinib or Sunitinib. And Avapritinib has been approved by the FDA also earlier on this year, specifically for patients with tumours harbouring the PDGFR alpha D842V mutation. And from the phase 1 trial, the response rate was a fantastic 91%. In terms of toxicity, neurocognitive side effects are something to look out for with this drug. But in the majority of patients, these side effects are manageable with again, dose reduction, dose interruption, and schedule change. Of course, earlier on this year, the Voyager trial that randomized patients to receive either Avapritinib or Ripretinib as third or fourth line therapy, the results became available, and there was no difference no significant difference between the drugs in terms of median progression-free survival. But notably, the response rate for the Avapritinib arm was 17%, and for Ripretinib 7%. So this drug clearly has activity not only for tumours that have the D842V mutation, but a subgroup of patients with tumours harbouring other PDGFR alpha and KIT mutations. But currently, we don't have a way of identifying those patients. Patrick Shofsky and the URTC have recently published the results of a phase 2 trial of another tyrosine kinase inhibitor, Cabozantinib, in recurrent GIST. And this trial met its primary endpoint in that 30 patients had tumours that were progression-free at 12 weeks. There were 7 partial responses, and 34 patients had disease stabilization, and the median progression-free survival was 5.5 months. And again, the side effect profile of this drug is similar to other tyrosine kinases. You notice diarrhea, palmar plantar hands, foot reaction, fatigue, high blood pressure, weight loss, and mucositis. And again, highlighting the point that it's important to monitor toxicity closely. Many patients require dose reductions and dose interruptions. So there's another interesting drug in development, PLX9486. This is a potent and selective inhibitor of the KIT D816V mutation. And a phase 1/2 trial in combination with sunitinib reported an impressive median progression-free survival of 11 months. And this is likely to lead to a future clinical trial. There are other more novel approaches using immunotherapeutic approaches. So this is an agent that basically is injected into tumours and has been investigated in metastatic renal cell and hepatocellular carcinoma. Recently, the results of a single-arm phase 1 trial were in advanced GIST was published, and basically this trial involved the use of this allogeneic inflammatory dendritic cell injected into these cells injected into the tumour with ongoing treatment with tyrosine kinase inhibitors beyond second line. And one patient on Ripretinib had stable disease for 9 months. Another patient on second-line sunitinib had stable disease at 12 months. These 2 patients actually had TROI partial responses. The median progression-free survival was 4 months, and there were no severe adverse events due to this, this agent. So again, a very exciting approach in advanced GIST. And of course, there's also a randomized phase 3 trial of another tyrosine kinase inhibitor in patients with advanced PDGFR alpha D842V mutated GIST. And this trial is currently running, aims to recruit about 120 patients, and patients are randomized in a 2-to-1 ratio to receive either Crenolanib or placebo. So, in conclusion, I think that the off-label use of various drugs in advanced GIST is a good option for select situations. And as I mentioned, this requires careful discussion between an individual patient and the oncologist. There's no sort of textbook answer to this, and it's a case of weighing all of the options up with individual people, including conventional treatment options as well as potential clinical trial options. Second point is that with NTRK inhibitors, I think we have very little knowledge currently regarding the frequency of NTRK fusions in sarcomas in general, but clearly for patients with tumours that do harbour these abnormalities, there have been impressive and durable responses. And thirdly, for clinical trials, I think that these are essential and very, very important for improving treatments and gaining access to further treatments and therefore improving outcome. So I'd like to thank all of the patients and the staff at the Royal Marsden and the ICR, and I've probably either put everybody to sleep or totally confused everybody. So that's why I thought it'd be good to have more time for questions at the end. So thanks very much.
Thank you, Robin.